We will be starting with the USMLE preparation, and first we will be covering up the first aid. In this channel there will be access to all the first aid explanation of every chapters. Each system will be covered in a day. Parts of this video will be uploaded in this channel only. Please subscribe for getting access to all the videos. First, we will take up the reproductive system, and in this reproductive system, the first thing to be discussed is about the reproductive embryology. First is about what are the important genes of embryogenesis. There are 4 important genes of embryogenesis. First is the sonic hedgehog protein. How can we understand or we can remember the functions of the sonic hedgehog protein or the gene? The sonic hedgehog is similar to that of an hedgehog. For example, the spines which stick out from the body of a hedgehog. This spine has a base, which is the zone which determines what is the base of the spine and what should be the tip of the spine. So, it has a polarization activity. It is involved in patterning along the anterior and the posterior direction. So we can say that it is associated with directioning along anterior-posterior direction, and it is helpful in determining the development of the base of the limb, which will determine the polarization in the anterior-posterior axis. It will also help in the development of the central nervous system. Any mutation in the sonic hedgehog gene will cause holoprosencephaly. Second gene is the WNT7 gene. This WNT7 gene, how we can remember that? It is present at the distal end of each developing limb. We have already studied that the base is determined by the sonic hedgehog gene. The tip is determined by the WNT7 gene. This tip is nothing but the derivative of apical ectoderm. How we can remember that? The base of the limb is covered. Rest is the length of the limb and the tip of the limb. The tip means apical and the fingers are ectodermal in origin. So we can say that this apical ectodermal ridge, it's produced by the development of the WNT7 genes, which will help in polarization and development of the distal end of each developing limb buds. What is the use? It is used for the proper organization along the dorso-ventral axis. Third gene is the FGF gene. FGF gives the idea of fibroblast growth factor. Fibroblast is present in the connective tissues in the extracellular matrix. These are all the remembering tips. How you can correlate and remember everything. And any kind of muscle is associated with hypertrophy and hyperplasia. So, this will induce mitosis. So, this FGF gene is also produced at apical ectoderm, but it will stimulate mitosis of underlying mesoderm. Thus, it will provide an axis for the lengthening of limbs. The fourth kind of gene is the Hox gene, also known as the homeobox gene. Box are always arranged from the top to the bottom if you just place it. So we can say each separate boxes, so we can say it's segmental. From the top to the bottom, so it is in a craniocaudal direction. And since it is in a craniocaudal direction, any mutation in this Hox gene will result in appendages or the limb in wrong location. For example, if the thoracic segment is associated with production of this upper limb, if there is any mutation, then this thoracic position of this spinal cord will not have upper limbs at the same position. So, the appendages or limbs will be present but in abnormal or wrong location. It's very important for the proper positioning of the appendages in craniocaudal direction. Hox gene is also important for protein transcription. It codes for transcription factors, some initiation factors in the process of transcription. So this was about the important genes of embryogenesis. We studied about 4 genes: sonic hedgehog, base of the limb, polarizing activity, anteroposterior direction, helps in CNS development, mutation results in holoprosencephaly. WNT7 gene present at the apical ectoderm, present at the distal end of the limb, arranging in dorsoventral direction. Fibroblast growth factor, lengthening of the limb, mesodermal mitosis, it's also produced at apical ectodermal ridge. Number 4, homeobox gene, box, segment, organization, top to bottom, craniocaudal direction, Hox mutation, appendages in wrong position, acts as a coding factor of protein or DNA transcription. This was about the important genes of embryogenesis. In next part, we will study about the early fetal development.